So when I describe gene therapy to like my patients or their parents for the first time, I explain fundamentally what sickle cell disease is, if we're discussing gene therapy in the context of sickle cell disease, which is predominantly what I do. And I say, you remember that sickle cell disease comes about because a person has inherited a portion of their hemoglobin molecule that's different. And that gene that gives rise to that hemoglobin molecule is made up of a bunch of letters. And in their particular circumstance, there's a misspelling of that particular gene. And that misspelling of the DNA code alone gives rise to their disease, the sickle cell disease. And so when I introduce gene therapy, I begin at the basics of what gives rise to sickle cell disease. And then I explain the different gene therapy techniques that are available under clinical trial investigation right now and how they're trying to correct that misspelling. There are multiple different strategies being used to employ gene therapy in sickle cell disease right now. They include treatments that can actually revert the spelling of the gene back to the normal way. Okay, that's called gene editing. There are gene therapy strategies that say, we're not worried about the misspelled gene that this particular patient has because we're going to introduce a new copy of that gene that's spelled in the correct way so that the patient can start making the correctly spelled gene and make the correct hemoglobin. And then there are other techniques that are a little bit more complicated, but they insert genes that suppress another gene. And then in so doing, it allows something called fetal hemoglobin to start to be expressed within the body. And that's important because fetal hemoglobin, as many of us know, is protective in individuals with sickle cell disease. So if you can get that level high, you can in many ways dampen and hopefully cure the disease altogether.